Hi, everybody.
Okay. Is it good? Thank you.
We just started.
So, as I was saying, tetanus is a deadly disease caused by a clostridial toxin, and it is produced and released in necrotic wounds on the skin. And the toxin is uptaken at the level of the neuromuscular junction by cholinergic nerve terminals. But instead of acting in here, the toxin is retro-transported to the soma of the motoneuron in the central nervous system. And here it is released and uptaken by its final target, which are GABAergic and glycinergic inhibitory interneurons, which are fundamental for physiological fact happening in our body. That is the control, that cycle of contraction and relaxation of opposite skeletal muscles. So in these interneurons, the toxin finds its final target, protein target, is the snare protein VAMP. The toxin cleaves VAMP, thus blocking the neurotransmission and causing the uncontrolled and hypercontraction of opposite skeletal muscles, thus leading to the well-known spastic paralysis of tetanus that can evolve in the arch position of tetanus and lead to death of patient by respiratory failure. So there are 4 types of the disease. Local tetanus, which is characterized by spasms nearby the site of the wound and the infection. General tetanus, tetanus neonatorum, and cephalic tetanus, which is a particular type of local tetanus localized nearby wounds of the head and the inner ear. So cephalic tetanus is a very aggressive type of tetanus with a very poor prognosis. And this is due mainly to 2 peculiarities of this type of disease. The first one is the first symptom, which is a non-canonical flaccid paralysis, so the opposite of what is expected from tetanus, that leads to a misdiagnosis of the disease and a loss of important time for the patient. The second one is the rapid progression of the disease. So there is the respiratory failure many times even before the appearance of the spasticity. So the patients are dying without even knowing why they are dying. But despite the importance of these 2 features, till a couple of years ago, there were no explanation about the mechanisms behind them. So that's why we decided to study deeper in this direction. And to do so, we first set up a model of cephalic tetanus by injecting the toxin in one of mice whisker pad to then evaluate the whisking behavior of the animals. And as you can see from this video frame, we observed the paralysis of the injected whisker pad already at day 1, lasting until the end of the experiment, and a progressive paralysis also of the contralateral side, which looked different from the one of the ipsilateral side. So to better understand these 2 types of paralysis, we applied the compound muscle action potential analysis. So we studied the efficiency of muscle contraction. And as you can see here from these peaks, from these peaks, we observed a great decrease in the CMAP in the injected side, which is very similar to that observed for botulism. So the flaccid paralysis of botulism. These results together told us the goodness of our model and also gave us an idea. Is it possible that in this case, tetanus toxin is behaving like botulinum neurotoxin, so on the other clostridial toxin? So to try to answer to this question, we first characterize a tool that was fundamental, which is an antibody to use in immunofluorescence and Western blot that detects VAMP, but only after its cleavage by tetanus toxin. So it's like a smoking gun proof of the activity of the toxin, and we were able to follow tetanus along its journey. And what we did was to cryoslice the whisker pads of the animals and check in immunofluorescence for cleaved VAMP. And as you can see here, we observed a great cleavage of VAMP in the injected whisker pad at the level of the neuromuscular junction, and instead, no sign of cleavage in the contralateral side. So thanks to these results and the previous one, we were able to answer to our first question and to understand that the flaccid paralysis observed in cephalic tetanus is due to a non-canonical peripheral activity of the toxin. So next, we move to our second question. So why is the progression of the disease so fast? And to do so, we move to the evaluation of the central effect of the toxin. And we focus on the brainstem. We focus on the brainstem because it contains not only those nuclei involved in the control of facial muscles, but also many nuclei which are fundamental for vital function like swallowing, respiration, and the control of heart rate. So we wonder if it was possible that tetanus toxin may act also on those nuclei in cephalic tetanus. And to do so, we did a huge, trust me, a huge screening of brainstem cryoslices. And in my opinion, the most interesting results are in this panel, because here the first line shows the cleavage in the para-gigantocellular reticular nucleus, which has a role in the control of respiratory rhythm. Hypoglossal nucleus and nucleus ambiguus, which are mainly involved in the control of swallowing, and also in the trigeminal motor nuclei, which have a role in the control of the respiratory upper airways. And these results were corroborated by great defects in ventilation that we observed in intoxicated animals using a method that we developed in our lab. So altogether, these results gave us the opportunity to understand that the progression of the disease, this rapid progression, is due to a huge activity of the toxin in the central nervous system in nuclei fundamental for vital functions. So this is a never-ending story, because we observed something, in my opinion, interesting that is reported here in the first line, because this nucleus doesn't have any connection to the periphery. So we wonder, how is it possible to see such a huge activity of the toxin in a nucleus that doesn't have the possibility to uptake the toxin and bring it back inside of itself? Is it possible that there is a wider spread of the toxin than what is reported in the literature? So to test this hypothesis, we slightly changed our model of intoxication. So instead of injecting toxin in the whisker pad, we injected it in between the lal muscles, so those muscles controlling the movements of the ears, which are again controlled by facial nuclei, but from different subnuclei of those controlling the snout. And we did this intoxication in animals that previously underwent the axotomy of one of the two facial nerves after the bifurcation that divides the innervation of the lal from that of the snout. So we maintained intact the communication between the facial subnuclei of the lal and the periphery, and we disconnected the facial subnuclei of the snout from the periphery. In this way, we were able to study the spread of the toxin inside the nucleus of the central nervous system. And as you can, I hope, see here, in the intact site, we observed a huge cleavage of VAMP, so thus denoting also a peripheral spread via lymph and via blood of the toxin. But what is more interesting is that in the axotomized site, we observe the cleavage of VAMP in the intact subnuclei, but also in those subnuclei that were disconnected from the periphery, thus denoting a central spread of the toxin wider than what was reported in the literature. And these results doesn't apply only to cephalic tetanus, because we observed a similar behavior of the toxin also in a model of local tetanus after the single muscle injection in the soleus. And as you can see from this rainbow color image, there was like a radial spread of toxin activity from those nuclei which uptake the toxin to other nuclei innervating muscles also far from the soleus. These results are important, in my opinion, also from a translational point of view, and I'll tell you why with a terrible rugby metaphor. So, when patients without a proper anti-tetanus vaccination goes in the ER with a risk of tetanus, what is usually done is the shot of anti-tetanus immunoglobulins. The problem is that once tetanus, that here is the rugby ball, is inside the motor neuron, that here is Johan Lomu, ticks are useless. It's too late. But if the toxin are going along these long journeys also in the central nervous system, is it possible for us to intercept, to block the toxin while traveling from one neuron to the other in the central nervous system and save those patients for whom the ticks are useless, unfortunately? So this is the direction we are, the path we are taking now. And we already characterize very potent monoclonal human antibodies, which are about 10,000 times more potent than ticks. And our hope is to use them to do the intrathecal treatment in those, in our case, mice, but in those patients for which ticks are not useful. It's too late to be used. And we are still working on this, so it's not the story for today. But our preliminary results are very promising, so but let's hope for the future. So I'm done with my presentation. I'd like to thank all the lab members and former members, collaborators, all my former lab members, and Professor Montecucco for being a great mentor in these years, and of course you for the attention.